Hi there, my name is Zach Wilson, and I'm going to be an instructor for one of the cell biology sections of the Foundations of Molecular Biology course, and that will be on September 28th, 2022. And in that class, we're going to look at how protein post-translational modifications regulate cellular processes, and I wanted to focus on one particular cellular process, and that's organelle inheritance. So the point of this video is to just connect those two ideas together so that in class we can really just delve into some experimental data looking at how protein post-translational modifications can regulate organelle inheritance, and we're going to do that in investigating one process in budding yeast. So right now I'm going to actually close down my video so that it doesn't block my slides. So I've noticed that happens in the past. Okay, so a quick outline of what's going to be covered in this pre-work video. We'll just start with a definition of protein post-translational modifications. And since that's a lot for me to say over and over again, you'll often hear me shorten that down to just PTMs. I want to cover some of the main regulatory roles of PTMs by how they affect protein characteristics or functions, cover two major actions and some beneficial features of protein PTMs that they provide to cells. And then I want to introduce organelle inheritance and specifically get to the point where we can examine how protein post-translational modification can regulate vacuole inheritance in budding yeast. At the very end of this video, and part of the slide set, is that I curated kind of overviews of 5 common PTMs. It's phosphorylation, ubiquitination, methylation and acetylation, glycosylation, and lipidation. And so really in thinking about PTMs, I think it's best to think about the cellular context and what they are regulating. So I didn't really think it'd be useful to just talk through each one of those, but I did think it might be useful to curate some figures for each of those PTMs. And then on those slides, I provided the pages within the molecular biology of the cell textbook that covers these different protein post-translational modifications. So I'll just quickly highlight those slides at the end, but I won't go much more into them. Okay, so protein post-translational modifications are covalent modifications to proteins. These are often reversible. So for example, in phosphorylation, you can have a protein that's phosphorylated by a kinase and then subsequently dephosphorylated by a phosphatase. And so then that gives you control of that phosphorylation event on that protein in both directions. And then proteins are often multiply modified, and that's highlighted in this figure from the textbook on P53, showing its multiple phosphorylation modifications at its N-terminus, and then a range of different modifications, including ubiquitin and acetylation at its C-terminus. However, there are some protein post-translational modifications that are irreversible. A common example to think about is proteolytic processing, and that's just shown with this example of insulin formation, going from the proinsulin form to its more final mature form after proteolytic processing and disulfide bond formation. So the main regulatory role of protein post-translational modifications is to alter protein characteristics or functions. And those characteristics or functions that are regulated, I've highlighted in this table with characteristics here on the left and different functions that are regulated on the right. So some of those characteristics that can change about proteins when they become modified, a lot of it is structural, so altering their folding or folding rate, their stability, or their conformation. And then another characteristic is dictating where they then localize in a cell, or if they associate with membranes, for example. On the functional side, protein post-translational modifications can dictate when a protein becomes active. They can alter enzymatic function. And even when I was creating this table, was thinking that activity and enzymatic function can be sort of confusing, but the reason I wanted to include activity is that, you know, there are proteins that aren't enzymes, right? And so they can become active in a different way. And you can also, activity is a common overarching term that you'll hear people use when they discuss protein post-translational modifications and how they're altering protein. Some other functions that they can control are what the protein interacts with. So controlling interactions with other proteins or other macromolecules such as DNA or RNA. And then finally, you know, the end lifespan of a protein. And when you think about that, maybe ubiquitination and the ubiquitin proteasome system, where that modification then leads to the turnover of the protein and hence the termination of its functional lifespan. So this table from molecular biology of the cell it again covers those same common protein post-translational modifications that I'm making slides of at the very end of this pre-work video. And what's highlighted in here is how these common PTMs can alter protein characteristics or function. So, right, for phosphorylation shows that, you know, it could drive the assembly of protein into larger complexes. But again, it's important when thinking about PTMs is thinking about them in the context of whatever cellular process you're investigating. But palmitoylation, which is a form of lipidation to proteins, that's a common addition that will then drive protein association with membranes. So it's an example of how protein PTM can alter protein localization. So one of the main things to think about with protein PTMs is how they can create this combinatorial control of protein function. And so depending on how the protein is modified and depending on the differential modification that occurs, this could lead to a regulatory code that then leads to a different functional output for the protein. This is very similar to what was covered also in gene expression, where, right, the combination of activators, enhancers, and repressors can provide this tight and exquisite control over how a gene becomes expressed. In the same way, how a protein is modified can lead to this exquisite control over what is the functional output of that protein, or where is that protein going to then go in the cell. In the text, they provide an example of this kind of regulation on the Src type kinase, and they use phosphorylation as that example where, right, multiple phosphorylation events are required before you then create an active Src type kinase. So one of them is actually the removal of inhibitory phosphorylation, phosphorylation on the activating ligand that Src type kinase then binds to, and then the addition of another phosphate group that leads to a conformational change, creating an active kinase. And they describe in the text, right, how this can then lead proteins to working as a microprocessor, where really the input of these different signals all have to occur to then get a specific functional output at the end. I think the other thing that's really cool and an important point to point out is that protein PTMs also now enhance the functional capacity of the genome. So in gene expression, talk about, you might go from gene X and then you could have different splicing variants to create different proteins from that gene, and they can have altered function. So now you have an enhancement from one gene into different proteins with different functional output. So you've got that enhancement there. But now the next step is that you can also now modify these proteins, and that can lead to an enhancement of the functional output. So it really starts to expand all the different capacity that a single genome could have, going down to the protein level and the vast array of functions that proteins can then have afterwards based on how they're modified. So beyond just this functional enhancement of the genome, there are two key beneficial features that protein post-translational modifications provide to cells. And one is shown here is that since the proteins are already made and you can just regulate their activity based off of quick modification, you can have this quick environmental response. And that's shown in this figure where you can have an extracellular signal that activates a receptor, and then through this just altered modification of proteins and protein function, you can quickly respond to that environmental signal. And that can still go through a response that involves transcriptional response, but you can't really respond as fast to what your environment is telling you if you always have to go through transcription to create the proteins and the functional output you need. And so PTMs can really allow cells to dynamically respond to their environment. The other key beneficial feature is providing spatial and temporal regulation of proteins and of cellular processes. And this was the main feature that I wanted to examine in class and use it as a way that we can look at experimental data and cover how investigators commonly look at how proteins are post-translationally modified and how that might affect a cellular process. And in that example, we're going to focus on vacuole inheritance in the budding yeast Saccharomyces cerevisiae. So budding yeast create daughter cells by budding a new daughter cell asymmetrically. So it's a polarized cell, and then they need to direct the transport of organelles into that daughter cell. And what I've done as part of your pre-work is I've asked that you read this short review on vacuole inheritance, and it's done by Lois Weissman. And this is an older review, so it's a review from 2006. But there's the point of having that older review is that, a lot of the common themes that are discussed in there have remained true for organelle inheritance in general, but also just the movement and spatial organization of organelles in the cell. And then also what we can do in class then is start from a point of where was vacuole inheritance research at a set time. So what we'll do in class is starting from 2014 and look at data that updates what is discovered about how vacuole inheritance is regulated through protein post-translational modification, covering data through multiple papers. So I think what might be a fair question that you may be asking is, hey, Zach, so why would we want to examine vacuole inheritance even in class, or why are researchers looking at it? And the reason that I wanted to focus on it and also highlights why it's been investigated is that when you look at this diagram of a eukaryotic cell from molecular biology of the cell, right? The eukaryotic cell is just has this elaborate organization of organelles that are interconnected and placed in certain spots that within the cell that can also be dynamically rearranged. And so that just creates this question of how is this elaborate organization established, maintained, and dynamically rearranged? And then with that, when you form a daughter cell that may need to be specialized for a different fate, how do you get organelles into that daughter cell, or how do you get determinate factors over into that daughter cell that alters what the fate of that cell might be? And so, when we look at vacuole inheritance in yeast, we can focus on this one particular process of taking, it's fairly simple organelle and considering how do you get this structure to go from the mother cell into the daughter cell. And in that, there's these questions that can think about or consider that the cell has to also address. And these same questions and the principles of getting the vacuole from mother cell into the daughter is shared in just organizing organelles or other molecules in the cell in general. So those questions are, where are you going? So how does the cell take an organelle or macromolecule to a particular spot? And what are the factors that are doing that? When are you going? So at what time to do that? And for organelle inheritance, right, that has to be coordinated with the formation of a daughter cell. And then importantly, when you get to a daughter cell, how do you know when you're there? And how do you end that transport of that organelle so that it stays put in the daughter cell? So what's covered in that review are the key protein factors that are involved in vacuole inheritance in yeast. But I'll talk about what these different protein factors are. But in particular, what I want you to notice is that you have a motor, right? A particular motor protein that will walk along the cytoskeleton. So that will give you directed transport. Often there's an adapter protein, so connecting a motor to a receptor that's on the cargo that's being transported. And then finally, you have a protein that's either an anchor or is the receptor, so that you know what cargo it is that is being transported. And then that's also either the cargo itself or it's tightly bound to the cargo so that the cargo can be moved from one particular spot to another. And so in vacuole inheritance, how that works is that the motor is myo2, so it's this myosin 5 motor that walks along the actin cytoskeleton. And myo2 interacts with this regulated adapter protein that's VAC17. And VAC17 connects the motor to the vacuole by binding to this vacuole anchor protein VAC8 that is multiply lipidated so that it's strongly associated with the vacuole membrane. And so the key protein in here, and the protein that we'll continue to study and look at in class with data, and where I wanted to get to so that we can examine data in class on this process, is this regulated adapter protein VAC17. And Lois talks about this in the review of some of the knowledge that is known about VAC17 at the time. And kind of highlighting how this is a regulated adapter for this process is that VAC17 starts to help address some of these questions of how the cell can time vacuole inheritance and also end it so that vacuoles end up in a daughter cell. And so on the where you're going part, right, that's a bit more on the motor, but the when are you going, they notice that VAC17 its synthesis is coordinated with the cell cycle. And in one slide, I'll show you what that data looks like. Importantly, to get it there, VAC17 assembly with VAC8 and myo2 initiates this process of vacuole inheritance. And then as part of the process of knowing how do you know when you're there, so how does vacuole know when it's in a daughter cell, VAC17, it was also demonstrated that its regulated degradation of VAC17 terminates vacuole inheritance. So this dark blue oval representing VAC17, it gets formed. It creates this assembly to start pulling vacuoles into a daughter cell. And then when they're there, it eventually gets degraded, and that helps to terminate the process of carrying vacuoles into a daughter cell. And so, this process is more complicated than just the synthesis and degradation of VAC17, because then you focus on VAC17 itself. Knowing when it is synthesized and when to precisely degrade it is tied to some regulators, and the discovery of those regulators of VAC17 is the data that we will discuss in class. So here's some of the data on how VAC17 is regulated in this process of vacuole inheritance. So on the top here is a Western blot. And what these researchers did is they took yeast, and you can coordinate when yeast go through their cell cycle. So you can stop a whole culture of yeast cells and such that they're all in G0 at your 0 time point, and then allow them to start going through the cell cycle. And in this case, samples were taken every 10 minutes as yeast went through their cell cycle. And for the most case, yeast end up ending their complete cell cycle after about 90 to 100 minutes. So when they followed VAC17 levels, and the part that is VAC17 is these upper bands. So VAC17 synthesis starts around 40 minutes into the cell cycle. It accumulates, and then you start to see its degradation, and it goes away in the culture, kind of completing the cycle of VAC17 synthesis and turnover. This is also shown down here with these fluorescence microscopy images. And so in green is VAC17 fused to GFP. In red is a different protein. We'll actually discuss what that different protein is in class. But you can see that VAC17 GFP, the synthesis of the protein and its turnover matches well with this Western blot data, where at the 40-minute mark, you start to see the synthesis of this protein. It's still there as the bud continues to grow. You still have VAC17 GFP, and then eventually it becomes degraded where you can no longer see it until the cell gets large enough that it divides away. So the other discovery that I want to point out, and this is leading up to where we're going to start in class, is on top here, and I'm showing the domain layout of VAC17 and some of its key domains. And what researchers found, and this is also discussed in the review, was that VAC17 contains this PES sequence. And what this PES sequence is it's defined for the amino acids that are enriched within this sequence. So proline, glutamate, serine, and threonine. And this sequence, it was known, often becomes phosphorylated, and then after phosphorylation regulates the degradation of proteins. And so they were curious if the PES sequence indeed regulates the turnover of VAC17. So they deleted this chunk, which is shown down here and highlighted in red. When they removed this area of the protein, there was strong accumulation of VAC17 compared to when you just had VAC17 expressed without any truncation to it. And then that's also shown here when they removed the PES sequence and looked at VAC17 when it's fused to GFP, and they see its accumulation. And importantly, they also see that it's still attached to vacuoles, but this seems to alter vacuole inheritance, where you can tell vacuoles are getting pulled from both the mother and daughter side right to this mother-daughter neck. So the termination of vacuole inheritance is also impaired when you cannot regulate the turnover of this protein. And so I brought this up a bit before, but this is where we're going to start in class. So this just highlights the exact amino acid sequence of the PES sequence of VAC17. And what we're going to do in class is we're going to examine data for how research went about examining the phosphorylation of VAC17 within its PES sequence. And we're also going to examine data for the role of an E3 ubiquitin ligase in and a localized kinase and their role in vacuole transport. And so that's where I wanted to get to, was just this primary goal of giving a little bit of a discussion on protein post-translational modifications, and then connecting that to vacuole inheritance and budding yeast, so that we can just, as class starts, examine some data on the post-translational modification of VAC17 and how that is studied to examine the regulation of vacuole inheritance. As I said, these next slides are curated overviews of 5 common PTMs. I'm not actually going to talk through them, but I will just briefly pull them up so that they can also be seen in the video. Thank you for your attention. So this next slide is on phosphorylation. There's some key pages. The text covers phosphorylation shown here. These next 2 slides are on ubiquitination. This is a slide on methylation and acetylation. Here are some pages where it discusses the role of these modifications in the histone code and the regulation of gene expression. This is a slide on different types of glycosylation, so N-linked or O-linked, and here's some pages discussing glycosylation in the text and their role in protein folding and stability. And then finally, lipidation, and here's the pages where it's discussed in the text, but main role of lipidation is to drive association of proteins with a membrane. And with that, I'm going to stop recording, and I want to thank you again.